So today I'd like to tell you about something that we're interested in the Crossley lab, which really has to do with getting to the bottom of the molecular basis of how genetic mutations result in human disease. And today I'm going to tell you about how having an understanding of this is really going to be of crucial importance as we head into an era of what I've called personalised medicine. So personalised medicine is essentially the notion that customised therapeutics can be tailored to an individual based on their unique genetic makeup. And really this stems from the idea that for a given disease might arise from an array of different and distinct mutations. And so a bunch of people could walk into a clinic with the same disorder, yet the way that you might feasibly treat each of those individuals could differ. And I guess the prospect of using widespread personalised medicine has really come to the fore in recent years due to the lowering costs of sequencing the human genome. So when the genome was first sequenced in 2001, it was done so at a cost of $100 million. But in 2007, there was a technological advancement known as next generation sequencing, which resulted in a drastic plummet in this cost. And shortly after, James Watson, the founder of the structure of DNA, he humbly had his own genome sequenced for a cost of $1 million. And in fact, these days, using one of these little benchtop machines here, just about anyone can have their genome sequenced for around $1,000. And in fact, this graph's a little bit out of date. And now they're actually even talking about the race for a $100 human genome. So in our lab, we breathe and live genes, I guess, but I realise that not all of you will be familiar with the structure of a gene. So I've put a little schematic up here, and it's shown in blue. And essentially, a gene is a stretch of DNA that has these functional units, shown in green boxes, that are sewn together. And you can think of a gene as a light bulb, really, in that genes are switched either on or off in different cells. And so a gene that's important for cognitive function is, I guess, hopefully switched on in a brain cell, but it might be switched off in your left knee or something. And what determines whether these genes are switched on or off are these proteins known as transcription factors. And I've used dimmer switches here to represent transcription factors. And what they do is they can either act in concert or they can antagonise each other to fine-tune the level to which a gene is switched on. So when it comes to genetic mutations, what you find is that mutations can either lie within the gene itself, and historically these mutations have been, in a sense, the sort of low-lying fruit of mutations when it comes to understanding how they work. So we have a very good idea of, when you identify one of these mutations, how it operates. But there are also mutations that lie outside of genes in these regulatory regions of DNA, and they are much more difficult to interpret. And the way that they work is they disrupt the binding of these molecular switches, and so the gene is not controlled appropriately. So these mutations that lie outside of genes, their detection is rapidly increasing due to all the sequencing that's going on, but it still remains quite a challenge to interpret the mechanism of operation. And so I'd just like to take you through a case that we've been working on in our lab, which is Haemophilia B. So this is a blood clotting disorder which affects roughly 1 in every 30,000 males. And this barrel of laughs here is Queen Victoria. And you may have heard of Haemophilia B before as the royal disease. And this is because descendants of Queen Victoria had a high prevalence of this disease. And about 4 years ago, it was determined that in the European aristocracy, the mutation lay within a gene known as the clotting factor 9 gene, or coagulation factor 9, and the mutation lies within the middle of the gene. And when scientists found this, they knew immediately how this mutation operated. But today I'm going to be telling you about some mutations that lie at the beginning of the gene in this regulatory region here. So I'll just zoom in now on this sequence of DNA, and this is it. And basically everywhere that I've written a number underneath the sequence, that's where a mutation has been reported. And you can see that there are these 3 clusters of mutations here. And all of these mutations are actually associated with a rare form of the disease known as Haemophilia B Leyden, in which boys suffer haemophilia up until around about the onset of puberty. And at that point, they spontaneously recover. And it's been known for quite some time that these mutations here disrupt the binding of a switch protein called HNF4. These ones here block the binding of a different switch protein. And together, all of these mutations operate by disrupting this gene being switched on. So you don't produce clotting factor, and that's why you bleed uncontrollably. And incidentally, the reason that boys that have these mutations recover is that at puberty, increasing levels of testosterone activate this other switch protein, androgen receptor, which can then switch on the gene. But today I'm going to be talking about this third cluster of mutations here, which for 20 years or so has been postulated to disrupt the binding of a third switch protein. And this is the guy that we've been going after. And just by looking at the DNA sequence here, we reasoned that there are roughly 2,000 switch proteins out there, but by looking at this specific DNA element, we reasoned that it might be a particular protein called one cut. And so we set about exploring whether this was this mystery elusive protein. And so the way that we do this is we take a DNA sequence that corresponds to this region and we label it with radioactivity so that we can detect it. We then synthesize this one cut protein in bacteria and purify it. We mix it together, and what we find, go back, is that the protein is able to interact with the DNA as shown here. So this suggests that at least in a test tube, this protein and this DNA can interact. But what we next needed to do was to show that the haemophilia mutations disrupt this binding. And so there are a number of different mutations in this region, and they differ with respect to the severity of the disease with which they're associated. And this is binding of the protein to just normal DNA. And when we look at this first mutation here, what we find is that it completely abolishes binding of the protein. And if we look at this least severe mutation, we find that it also disrupts binding, although not to the same degree. And these intermediate mutations here sort of block binding somewhere in between. So this was all looking pretty good, and it suggests to us that most likely this protein that operates through this site is one cut. But really all of these experiments done so far have essentially been done in a test tube, and we really need to show that the same thing is occurring in living cells. And so to do this, we collaborated with a group in Cambridge, and we used a technique known as chip sequencing. And essentially what that allows you to do is to look at where a switch protein binds throughout the entire genome. And what we found is that when we looked at this one-cut switch protein, we found that it is in fact at the beginning of the factor 9 gene. And we saw it there together with its other molecular switches, CEBP and HNF4. But what really clinched it for us, I guess, was when we collaborated with another group in Belgium who had a mouse model in which they deleted this one-cut switch protein. So here you've got a normal mouse on the left and a mouse on the right where this one-cut protein has been deleted. So it's no longer there. And you can immediately see that this mouse is unable to produce factor 9. And it can actually produce all of its other normal clotting factors, and its other switch proteins are also intact. So this effect really is specific and direct and due to one cut. So that's just one case that we've been working on in the lab. We work with other disorders as well, such as sickle cell disease. But I'd just like to, I guess, wrap things up now by just talking about some of what I think are the biggest hurdles in moving ahead with personalised medicine. And the first one that pops to mind really is that it's very difficult to discriminate disease-causing mutations from just naturally occurring genetic variation that exists in all of us. That remains very challenging. And we can sort of get to the bottom of this if we start to understand the complete suite of these human switch proteins. But this is no sort of mean feat. And there are 2,000 of these human switch proteins. And there are, depending on how you classify them, at least 200 different human cell types. And to make matters worse, these switch proteins behave differently in different cell types. So you can do the maths and work out that there's a huge number of permutations that you need to investigate. Not all switch proteins are found in all cell types, but nonetheless the number is still large. Fortunately though, there's a lot of research going into this, and perhaps the most notable of these is a large public research consortium known as ENCODE. This is a big international collaboration that's been going on for about 10 years at a cost of $400 million. And just last year they published a big study where they looked at 119 of these switch proteins across 72 cell types. So this really is only sort of scratching the surface of what we need to look at, but it has certainly delivered to us quite a valuable treasure trove of information that we can now tap into. And I guess on a positive note, working with these switch proteins is in a way like doing a jigsaw puzzle, and the more that you characterise these proteins, the easier it is for the final pieces of the puzzle to be put into place. And some of the things that help us are that these switch proteins, what you find is that you get switch proteins that belong to a single family, and once you've characterized one, you can then start making quite educated guesses on how other related switch proteins are also going to act. The other thing is that you start to see patterns that emerge. So I showed you before that these 3 switch proteins are all found at the beginning of this factor 9 gene, and you can see that they're also all found here together. And in fact, if you then extend that to not just looking within this gene, but looking across the whole genome, what you find is that these 3 switch proteins often co-associate. And if you zoom in on where that's happening, it highlights these critical regulatory regions that are often mutated in a range of human diseases that have to do with regulating sugar levels and fat levels in blood, and also other clotting disorders. So on that note, I'd just like to finish off by summarising and saying that in the Crossley lab, what we do is we investigate the molecular basis by which these complex mutations disrupt the activity of these switch proteins. And I've shown you how this cluster of mutations here disrupts the binding of this particular protein, 1-cut. And I think this is quite an elegant illustration of how all of these different mutations that essentially result in the same disease, they all do so by very different mechanisms. So they all disrupt different switch proteins. And understanding these complex mutations is certainly going to be a vast undertaking, but it really is going to be essential as we move forward for the efficacy of personalised medicine. And now it just leaves me to thank the lab, also our collaborators and funding bodies. Thank you.